Hello everyone. Here with a new topic. We're going to talk about the nervous system a little bit. And so we've got several things that we're going to talk about, and we're going to talk about them in some detail. So let's get to work. I want to talk a lot about neurons. And so these are the cells, the nerve cells that are central to the whole nervous system, and also synapses. And although this is kind of a small outline, both of these there's a lot of detail. So we're going to get to quite a bit of detail about both of these things. Okay, so big picture stuff. Organisms must sense and respond to their external environment. So we talked about the external environment a little bit when we talked about homeostasis and how, interactions with the external environment are what, we need to exchange things, and that's one of the reasons we need to maintain homeostasis. This is related to that, but basically it's a fairly obvious thing, is that organisms don't exist in a vacuum. And so you need to be able to sense the external environment and then make yourself respond to it. And so you need to do that by sending signals through your body. Multicellular organism, there are all kinds of different things to sense, and so there's all kinds of different signals that need to be sent to the brain and processed. And how does that signal gets transmitted? And then once the brain decides what to do, then other signals need to go to the muscles or whatever to affect a response. And so you need to pass that signal around the body. So the nervous system is used for fast signaling, where you can the message gets passed very quickly. The hormone system, or the endocrine system, that is for slower signaling, which is still pretty fast, but not nearly as fast as a nervous system. And so these two systems are important for signal transmission. Okay, so like I said, the central player in this drama is the neuron. Nerve cells are called neurons. And the basic idea of a neuron is it can take a signal from one end of the cell and pass the signal to the other end. And so where the signal starts is called the dendritic end, or the dendrite, and where the signal ends up is called the axon. Now, recall when we talked about muscle cells, how muscle cells are not typical cells. They're very long. Same thing with nerve cells. They're very long. And so by passing a signal from one end of a cell to another, that signal gets passed a relatively long way, right? These are not like typical microscopic cells. So here's a figure from your book kind of showing the general morphology of a neuron. And so the signal starts on the dendritic end. Dendritic means branching. And so if you take a class in dendrology, that's studies of trees, right? Trees have branches. Sometimes streams, we say they're dendritic because they've got lots of branches. And so the dendritic end, or the dendrites, that's the branching end. And so that's where the signal starts. And then the axon is where it ends up. So the signal starts at the dendritic end and gets passed to the other end, which is the axon. So now those dendrites, there's all kinds of things that can start the signal. They might be associated with a receptor, which senses the environment. Remember, we said this is all about organisms sensing their environment. So it might be something that, it might be smell, or taste, or touch, or temperature, or sound, or whatever, light. And so that might be associated with the dendrite to start the signal, or that dendrite might be associated with the axon of another neuron. And so that's how a signal can get passed from one neuron to another. And so again, that's something that's kind of interesting here. Not only can you pass a signal from one end of this long neuron to the other end, then you can transfer that signal to another neuron. And that's how, in a large organism like us, signals can get passed all over the body. You don't have a single neuron running from your brain all the way down to your big toe. You've got several of them, and they can pass the signal back and forth. And so that's basically the two things that can be going on at the dendritic end to start that signal. Now, once the signal gets to the axon, to the other end, then that axon might just link up with the dendrites of another nerve and just pass the signal to another nerve, or that axon might associate with what's known as an effector. So something that does an effect, something that responds to the environment, like a muscle. And so if you think back when we talked about how does a muscle fire, we said the first thing is calcium needs to be released to cause that muscle to contract. And we said, well, what causes the calcium to release? A signal from a nerve. Well, here you go, right? This is sort of what we're talking about. Okay, so the afferent neurons, or the sensory neurons, carry signals from the receptors to the central nervous system. So now, we're focusing more on vertebrates, and specifically, most of this is going to be directed at humans. The afferent, or the motor neurons, carry signals from the central nervous system to the effectors. And remember, we said A comes before E, afferent becomes before efferent. And so the signal starts with the afferent neurons, goes to the central nervous system. The central nervous system processes the signal and then sends the result to the efferent neurons to cause the organism to do something. So the sensory neurons, the motor neurons, those are basically the peripheral nervous system, as opposed to the central nervous system, the brain and the spinal cord. Now, the neurons that make up the central nervous system, these don't really carry a signal from a receptor or carry a signal to an axon. They just carry a signal from one neuron to another. And those are sometimes called interneurons, right? Within neurons or something like that. Just a little vocabulary. Okay, so when we look at neuron anatomy, some neurons, not all, but some, are covered by a myelin sheath. And so those cells are said to be myelinated. These myelin sheath is produced by specialized cells called Schwann cells. And the purpose of this myelin is it acts as an insulator. It does not carry a signal very well. And you say, well, that's odd, because the whole point of a neuron is to carry a signal. So why have these neurons that are covered with this insulator? Well, we'll talk about that in a second. But one reason might be the same reason you insulate wires in your house. You don't want signals going from one neuron to another neuron just because they're laying next to each other. But there's a more important reason, which we'll get into here in a second. A more important reason for that myelin, which we'll go into in a second. Okay, and so then you can have lots of these neurons that bundle together to make a nerve. So whenever we dissect something and we see that nice white, whenever you're dissecting, nerves are usually pretty easy to see in animals because they're very white. And that nerve contains many neurons in it. And so if you look back at this figure again, they're showing that this is a myelinated neuron. And you got that myelin sheath. And now if we're looking at a figure from your book and it's sort of showing a nerve and showing how you can have many of these neurons bundled together to form a nerve. So if you're familiar with a fiber optic cable, that carries your internet to and from your house or something like that, it's similar. You've got all these small wires, for lack of a better term. That would be a neuron that carries a signal, and you could put a bunch of those together and they can carry lots of signals. And you can see in this example how you have both myelinated and unmyelinated neurons. Now, we said that the whole point of a neuron is to pass a signal from one end to another. That signal is called an action potential. And so the action potential starts at the dendrites and then is propagated down to the axon, or it moves down to the axon. So again, these are all just vocabulary terms that you need to be familiar with. All right, let's talk about this action potential. What is it specifically? So we're going to look at this in detail like we did with the contraction of a muscle. And again, it's going to show by how understanding things at the molecular and cellular level, you really start to understand how they work. The basis of this action potential is an imbalance of ions across the neuron membrane. Okay, so there's a lot in that sentence. Break that sentence down, right? It's an imbalance of ions. First off, again, what's an ion? An ion is a charged particle, right? It's an atom that has a charge on it. Now, there are specific ions that are important to the action potential: sodium, chloride, and potassium. And you see that each of those has a charge. The sodium and the potassium have a positive charge. The chloride has a negative charge. And all this is going to be very important here. So this action potential is an imbalance of these ions across the neuron membrane. So again, the neuron's a cell, and it's got a cell membrane, just like we've been talking about all semester. And you've got an imbalance of these, meaning you've got more of an ion on one side than another. Well, think back to when we've seen this before. For example, electron transport chain, right? We were purposely creating an imbalance of ions. We were purposely pumping protons to one side of a membrane, hoping that they would, so eventually they would diffuse back, and as they diffused back, something would happen. This is the same idea. We're creating an imbalance of ions across a membrane so that when allowed, they will diffuse back. And that's the basis of an action potential. So again, it's the same fundamental ideas of like diffusion and ion channels and membranes. Now we're putting them to work, and we're understanding how we can put them together to see how an animal functions. So specifically, how are they imbalanced? The sodium and the chloride levels are relatively high outside the neuron. The potassium levels are relatively high inside the neuron. This is at the resting state. So a typical neuron that's just sitting there, that's not carrying a signal, this would be the conditions. Sodium and chloride high outside, potassium high inside. You also have something else that plays a part in this, are negatively charged proteins. So remember, proteins are big, right? Potassium, sodium, chloride, these things are all small molecules. We know that proteins are big molecules. And so, these proteins don't diffuse out of the neuron because they're so big. And in this instance, there's a lot of these that tend to carry a negative charge. And so they're going to be important when we're talking about the charge balance here. And don't forget, there's some negatively charged proteins that are playing a part. But they're only inside, they're not outside. Okay, so here's a figure from your book trying to kind of graphically summarize what I just told you. So take a look at this and study this. And you can, first off, big picture, you see the neuron, you see the dendritic end, you see the axon end. And then you see that they're zooming in on the membrane. So again, this whole thing is covered by a regular phospholipid bilayer. And we're going to zoom in on it. And they're showing you the inside of the axon and the outside of the axon. So the axon's not really the tip, it's the whole long part of the body in general. So just don't get confused by the terminology here. And so in these bar graphs, they're sort of showing you the relative amount of each of these ions. And so if you look on the left, it's showing you the relative amount of things inside the cell. And you see large impermeable anions, negatively charged. That's those large proteins I was talking about. You see there's a lot of them inside the cell. Now look at the chart on the right. The chart on the right is showing you the same ions, but outside the cell. And if you look at the large impermeable ions outside the cell, that's almost zero. There's none, right? So then again, look at the left chart. Potassium, big bar, very high inside the cell. Now look at potassium on the right chart, very low outside the cell. And then you see for sodium and chloride, it's opposite. So what they're just trying to show you the relative numbers of these things, like I just described. You also look at that zoom graph in the middle there. It says inside the axon is negative 70 millivolts, outside the axon is 0 millivolts. And so, again, you got to think back to what does voltage mean? Voltage means a difference in potential, right? How different the two sides are. And so if you talk again about a battery, a 12-volt battery, you've got a positive and a negative terminal, and each of those is connected inside that battery to different things. One is connected to lead, one is connected to acid, and they're not connected inside the battery. So if you ever ripped a battery apart, which don't do that. And so one of those chemicals in that battery really wants to give off electrons. The other chemical wants to accept electrons. And so if you connect those two things, the two poles with a wire, electrons are going to flow from one to the other because chemically they really want to. How badly do they want to? That's voltage. You know, how great is that difference in potential? And so, 12 volts, they really want to fly from one pole to the other. But in the socket in your house, you got 110 volts. If you go to where your dryer's plugged in, you got 220 volts. So those things so there's a lot more power, there's a lot more energy there. There's a lot greater difference in potential. Anyway, it's how badly, a difference in potential, if you listen to the term difference in potential, it's how different the sides are, okay? And so in this example, the inside of the neuron is at negative 70 millivolts relative to the outside. So we're talking millivolts. We're talking 1,000th of a volt. So obviously we're on several orders of magnitude different than your car battery or your outlet in your house. But the point being is the inside is negative relative to the outside. That's the important point here. But how different these numbers are tells you how different the charge is, all right? So these numbers will be important later on when we discuss the action potential in detail. That's all I want you to know. But I also want you to realize that at rest, the inside of the neuron is negative relative to the outside because of these ions. So that's another point I want to make. We've got this positive potassium inside. We've got this positive sodium outside. We've got a negative chloride outside and a negative protein inside. How come all these things don't just balance out? How come there's a difference in potential? It's because of the relative numbers of these that we're going to talk about. So even though there's a lot of positive potassium inside the cell, there's even more sodium outside the cell. That's why it's positive outside the cell. Even though there's negative chloride outside the cell, there's even more negative proteins inside the cell. You understand what I'm saying here? And so, on balance, the net effect of all these things, something just bit me. That really hurt. So I think it's going to rain here, and sometimes the flies start biting. That really hurt. Sorry about that. Anyway, because of all the relative numbers of these ions, the net effect is the inside of the neuron is negative relative to the outside. All right. Now, side note, again, it shows here the anions have a negative charge and the cations have a positive charge. So the way I remember this is cations have positive, right? Like a cat has paws. So take it or leave it. Okay, so if you look at these relative numbers of these, or the relative concentrations of these ions, like for example, potassium, where does potassium want to go, right? It's very high inside the cell, it's very low outside the cell. So simple diffusion says what? Potassium wants to leave the cell. Again, it doesn't want to, but it's energetically favorable. You know how diffusion works. By random molecular movement, there's going to be a net movement of potassium from inside the cell to outside the cell, if it's allowed to. But we got that membrane there, right? We have that membrane that doesn't allow potassium to diffuse out, although it wants to. Where does sodium want to go? Sodium's very high outside the cell, very low inside the cell. Sodium very much wants to go inside the cell, but it can't because there's a membrane there. And so what we're talking about, that negative 70 millivolts, that's called the resting membrane potential. This is a neuron at rest that is not propagating an action potential, that's not sending a signal. Well, how does it get that way? How do you get this, it's more positive outside than inside? Remember the sodium-potassium pumps. We talked about these in a couple of lectures already. That there's these very common pumps that use ATP, and they pump sodiums out and they pump potassiums in simultaneously. And for every 3 sodiums that get pumped out, 2 potassiums get pumped in. Well, there you go, right? So if you're running these things and you're burning ATP, and every time you push 3 positives out, then 2 come back in, that's a net of 1 positive every time that pump runs, right? That's how you can make the outside more positive relative to the inside, because there's a net export of those positive charges because of the sodium-potassium pump. Now, like I said, that sodium really wants to come back into the cell. And for the most part, it can't. The membrane stops it. But membranes aren't perfect. And in this example, this membrane is a little bit leaky. And so even though you're pumping all those sodiums out, some still leak back in. And the neuron needs them to be out. So you spend more energy. You burn more ATP to pump those back out, and a few leak back in. Most of them stay out, a few leak back in. So you're constantly running these pumps. And again, there's something like 40% of the energy you ingest is just to do this sort of thing. I don't know, don't quote me on that. But a huge amount of the energy you ingest is to run these pumps. Because if you don't have this difference of potential, the nerve doesn't work. This is what happens when you're brain dead, right? If you're suffocating, or if you run out of oxygen or whatever, and you can't make ATP, then you can't run these pumps, then the neurons in your brain can't establish this resting potential, and once your brain can't work, then you're dead. So these things are kind of important. But again, why do you have to eat 3 meals a day? You need energy just to run these pumps, just to keep your neurons ready to go. Now, we still haven't passed a signal yet. This is just to get the neurons ready to pass a signal. So this is a figure going all the way back to chapter 3 again. But again, if you walk through the steps, this is just a protein embedded in the membrane of the neuron. And 3 sodiums bind to the protein. Then ATP comes along. The energy from the ATP transfers a phosphate, which transfers the energy, which causes that protein to change shape, which ejects 3 sodiums, but simultaneously grabs 2 potassiums. And then as the potassiums bind, the enzyme changes shape back to its original shape, and it throws those potassiums in the cell. And we're back where we started. Remember, enzymes catalyze a reaction. They make a reaction more likely to happen without themselves being changed. This is what you're seeing here. And the net effect is you're pumping more positives out than you're bringing in, so the inside of the neuron is negative. And so this goes back to what I was saying earlier. The resting potential is negative 70 millivolts. Inside is more negative relative to outside. And again, voltage is one of those things like osmolarity. You have to be comparing 2 things. And so here we're comparing the inside to the outside, and it's more negative on the inside. Okay, so that's the resting potential. That's the neuron sitting there ready to carry a signal. So how does it carry a signal? How do we start an action potential? So remember, we said that the sodium is outside and it really wants to come inside. Well, we've had this situation before, right? Where we've had a high concentration on one side and a low concentration on the other side of something like protons in the ETS or whatever. So how do we solve that problem? We open gates. And you open protein channels that allow the sodium to run into the neuron. And so once those gates open up, now the sodium can rush in because there's a, the sodium is very high outside and very low inside, so it wants to diffuse in very rapidly once that gate is open. Well, how do you open that gate? Those gates are voltage sensitive. And so if the voltage changes, that causes the gate to open. And so if the voltage changes and the gate opens, then sodium rushes in. And so at this point, we call this depolarization. So when the membrane is at rest, it's polarized, right? Remember we talked about a polar molecule? What was a polar molecule? Where the charge is separated. The negative and the positive are apart from each other. At a rest, the neuron, what do we have? The charges are separated. The negatives are on the inside, the positives are on the outside. That membrane is polarized. When the sodium rushes in, now it becomes depolarized. Now you no longer have positives on just one side, you have positives on both sides. And so this is called depolarization when the sodium rushes in, and it's the start of an action potential. So here's a figure from your book kind of showing you this. And so you look at the far left, and you look at this nerve, and we've got two membranes, right? Both sides of the nerve we're looking at here. And you see on the far left where I've indicated, the membrane is polarized. You've got negatives on the inside, positives on the outside. That's the resting potential. That's a neuron at rest. And it's about negative 70 millivolts inside relative to outside. But then you see that a little bit further down toward the middle of this picture, you've got a sodium channel open. And so the sodium channel is open, and they're showing the sodium is rushing in. Those positive sodiums are rushing in because their gate got opened. And so at that point in the membrane, the potential is now plus 35 inside. So now all of a sudden, so much positive charge runs in, you go from a negative 70 on the inside to a positive 35. A huge swing in the charge and a huge change in voltage. But what do we say? These gates are voltage sensitive. And so when you change the voltage, you open other gates. Well, this is how we change the voltage. So now, when that voltage changed because the sodium rushed in, that caused a change in voltage from negative 70 millivolts inside to plus 35. Well, what's going to happen to nearby sodium channels? They're voltage sensitive. They sense that change in voltage and they open up. So what happens? Sodium rushes in there. So now you have another, at that part of the membrane, the voltage changes from negative 70 to plus 35. Well, what happens to sodium channels that are a little bit further down the neuron? They sense the voltage change, they open and allow sodium to rush in. And so you can see how that signal can get propagated down the length of that neuron. Because once you allow some sodium to rush in, that changes the voltage of that little part of the neuron, but it also opens nearby sodium channels, which changes the voltage of that little part of the neuron, which opens nearby sodium channels, which changes the voltage of that. And so that signal just moves down the axon because of this change in voltage and because these gates open in order from the dendrite down to the axon as that sodium rushes in. And that's the, now we've got a signal. It's being passed from one end to another. So again, why did the voltage change? Because sodium rushed in. And that caused a change that causes the next gates to open, so more sodium rushes in. Which causes the next gates to open, so more sodium rushes in. Is this a positive or a negative feedback loop? I'll give you a second. So our equilibrium, was negative 70 millivolts inside, and that was the resting potential, and the cell was resting there. Once we started this, once we changed the voltage in one part of this neuron, then that voltage, then that opened gates, which opened more gates, which opened more gates, which opened more gates. And so once you started it, you pushed away from that equilibrium, didn't you? Once you started this signal, instead of going back to where you started, you just kept going, right? That's a positive feedback loop. And so here's an example of a positive, and what do we say about positive feedback loops? You know, once you start them, you can't stop them until something else happens. And what happens is you reach the end of the neuron, right? And then something radically changes, right? But you don't go back to where you started. Well, now you can imagine, look at this figure here, you can imagine how this action potential can move from one end of the cell to the other. All you have to do is open channels on one end, which changes the voltage on one end, but then that opens nearby channels, which changes the voltage, which opens nearby channels. And so once you start it on one end, then that signal is going to propagate all the way to the other end. And so if you look at the charges here, you see how the charges are negative inside and positive outside, but then as those positives move in, that changes the voltage and more positives move in, and that signal can move all the way down the length of the neuron. Okay, so an obvious question is, what starts it? You know, once we open that first gate, then it takes off. You know, it's kind of an all. Once that first gate opens, the sodium rushes in, then boom, there goes the signal. Well, what opened that first gate? How did we start this whole thing? Well, we'll talk about that coming up. We'll get into more details about how we open those first gates. But the important thing now to realize is once you get those first gates to open and that sodium to rush in, then that can open other gates. And once that happens, then the signal just keeps going because you just keep opening gates all the way down. All right. Now, once you've passed the signal, you want to use this nerve over again, right? This is not a one-shot deal. You need to repolarize the membrane. Remember, the membrane was polarized. We depolarized it by allowing those positive sodiums to rush in. Now we need to repolarize it. We need to get back to where we started. And then once we do that, then it's ready for the next signal, right? Okay, so the first step to repolarization actually involves potassium, not sodium, okay? There are also potassium gates, and those gates are also voltage sensitive. But remember, potassium is high inside the cell. Potassium wants to leave the cell, right? But it can't because its gates are closed until the voltage changes. So in the first part of this figure, you see you've got a potassium gate that's closed. So that potassium really wants to leave. When the voltage changes as that action potential moves through, now that potassium gate opens up, now that potassium can leave. Also, think about what else is going on here. First off, potassium really wants to leave already. There's lots of potassium in the cell. There's none outside the cell. Now we're letting a bunch of sodium come in, which is changing the voltage, and the sodiums have a positive charge, right? The potassiums have a positive charge. What do same charges do? They repel. So not only is there a high concentration of potassium inside that neuron, but also now there's a lot more positive charges, which also makes that potassium really, really, really want to leave. And so once that potassium gate opens, that potassium goes flying out, right? But that gate won't open until it senses a change in voltage. But when the action potential comes through, those sodiums cause a change in voltage. Pretty slick. So now those potassiums run out, but now, excuse me, I had a frog in my throat. Because those potassiums really, really, really want to fly out, a ton of potassium flies out. So much that the voltage drops back to where it once was, or very close, what happens to the sodium channels? This causes the sodium channels to snap back shut, right? They opened when it became more positive, when they sensed that positive change in voltage, but now because all those positive potassiums run out, now we're sort of back closer to that negative 70 millivolts, those sodiums close. And so the sodiums can't diffuse anymore, and the sodiums get trapped inside the cell. And a bunch of potassiums just ran outside the cell, right? Well, to get back to repolarize the membrane, to get back to where we started, we need to flip-flop that. Well, how do we do it? Well, luckily we've got sodium-potassium pumps, right? So the sodium-potassium pumps kick in, they pump out 3 sodiums, bring back in 2 potassiums. They burn a lot of ATP to do it, but once they do that, now we're back to our resting potential. Now we're back to the negative 70 millivolts inside and lots of sodium outside and lots of potassium inside after those pumps kick in. Okay, so let's just review the steps again real quick. At rest, the neuron's got a lot of sodium outside of it, a lot of potassium inside, many negatively charged proteins inside. So the net effect is that the inside of the neuron is negative 70 millivolts relative to the outside, and the membrane is polarized. When you open up sodium channels on the dendritic end, sodium rushes in because there's a high concentration of sodium outside. Those sodiums, because they have a positive charge, now change the voltage. Now the inside of the cell is positive 35 relative to the outside, and the membrane is depolarized. Well, this change in voltage causes nearby sodium channels to open, which allows more sodium to rush in further down the nerve, which causes voltage to change, which opens other sodium channels further down the nerve. And in this way, that depolarization event, which started on the dendritic end, that action potential moves from one end to the other. And so this is why, you have directionality in nerves, right? The signal does not travel in both directions. The signal always travels in one direction, and it always starts at the dendritic end, and it always moves to the axon end because of the way these channels are arranged. Once that action potential is passed, we need to repolarize the membrane so we can use the nerve again. The first step is to open channels for potassium to run out, and then sodium potassium pumps kick in to reestablish our resting potential, which is lots of sodium outside, lots of potassium inside, negative 70 millivolts inside, because you pump out more sodiums than you pump in potassiums, and because you've got all those negatively charged proteins and things. Okay, so here's a figure from your book, kind of just again showing you that propagation of that signal over time and how that signal moves down the neuron, and you can kind of study this and see it just is graphically showing what we've been talking about, how you start at negative 70, then the potential goes up to plus 35, and then it gets reestablished back down to negative 70. Okay, well, let's go back to the myelinated neurons. In the unmyelinated neuron, that's kind of what we just talked about. That action potential just moves smoothly down the entire length of the axon. Great. But in a myelinated neuron, that myelin acts as an insulator, and you can't propagate the signal where that myelin's sheath is. And instead, you've got gaps. So you've got some myelin in a little gap and some myelin in a little gap, which is what they're showing you here. And so the signal, instead of moving smoothly along the length of the axon, it jumps from gap to gap to gap. And it's called, I think, saltatory, right? Saltation is jumping. And so that signal jumps over the myelin sheath and goes to a gap, goes to a gap, goes to a gap. Well, it turns out that this is actually much faster than if you just allow the signal to travel down an unmyelinated neuron. And that seems a little confusing that the insulated neuron is actually faster than the uninsulated, but I think the reason it's faster is because you can concentrate the sodium gates and the potassium gates at those gaps. And so the ions, you've got a higher concentration of ions, and so they move in faster. I'm not 100%, I don't 100% know the physiology and anatomy of this, but I do know that myelinated nerves carry signals much faster. And so nerves that have to work really, really fast are covered in this myelin sheath. And this is why. And so that's what I'm saying here, right? Is that myelin acts as an insulator, which causes that charge to jump from gap to gap, which makes these faster. Okay, so now the one ion we have not yet talked about is chloride, right? And so if you look here, you remember that chloride is very high outside the cell and very low inside the cell, okay? Now, you recall in one of our labs we talked about roundworms, so the phylum Nematoda, the nematodes, and we talked about a common roundworm that infects dogs is heartworm, and you should be giving your dog heartworm preventative, right? And the preventative, that pill that you give them every month, kills the juvenile heartworms. If the dog has adult heartworms growing in their heart, you need other medicine, and it's a very tricky thing. That's a very tricky treatment to get rid of those heartworms safely, those adults. But once you get them on the preventative, then if they get infected by a juvenile, that preventative will kill the juvenile, and then they'll never develop the adult heartworms. But how did that preventative, how does it kill the juveniles? It binds to chloride channels in the nerve cells and holds them open. And that leads to the death of the juvenile roundworm. All right, now that you know how this nerve works, why does leaving chloride channels open kill the juvenile roundworm? So think about it. Chloride's really high outside the cell, really low inside. If you hold that gate open, what's chloride going to do? It's going to rush into the nerve cell. All right? Now, the roundworm can pump the chloride, can use ATP and pump that chloride back out, but it's just going to diffuse right in. So that chloride's just going to keep getting into the cell. So remember, a typical nerve cell has a resting potential of negative 70 millivolts inside. It's relatively negative inside. Well, what happens to that resting potential if you add more negative chlorides? It's negative inside the cell. You're bringing in more negatives. It's going to get even more negative, like negative 100 millivolts. So now the inside of the neuron is even more negative because those chloride channels are held open. Okay, well then remember, how does a nerve propagate that action potential? When you open the first sodium channels over at the one end of the cell, sodium rushes in. And typically, that sodium rushes in and it brings the resting potential, it depolarizes the membrane, it brings the inside of that cell from negative 70 up to plus 35. That voltage change triggers sodium channels that are right next door, and they open. And then they open the next ones, they open the next ones, right? But what if the inside of that cell was very, very negative, negative 100 millivolts or something? Now you open up those first sodium channels, the sodium still rushes in, it still raises the voltage, inside the cell becomes more positive, but it doesn't become positive enough to open up nearby sodium channels. So even though you open up those first sodium channels, they don't trigger an opening of the nearby sodium channels. So the signal doesn't propagate. So we call this hyperpolarization. That the membrane becomes very, very polarized. It's negative 100 millivolts, or I don't know exactly the millivolts, but it's about a negative 100, 110. And so when sodium rushes in, when you try to fire that neuron, the signal doesn't get propagated. Well, if nerves can't propagate signals, then the organism can't live, and that's how you kill these juvenile roundworms. So again, by sort of understanding the basic function of this, you can start to understand a lot of other things. All right. Well, the next topic we need to tackle now are synapses. You remember we said that nerves can pass a signal to another nerve. So when that signal gets to the end of the nerve, it needs to pass that signal on. There's no point in carrying a signal from one end of a nerve to another if it doesn't do something. Remember, that's the whole point, is signaling and carrying information around the body. And so you need to either pass that signal on to another neuron, or you need to pass that signal on to an effector, like a muscle or something. So just getting the signal to the end of a neuron isn't good enough. You got to do something with that signal. Well, where the neurons meet and transfer that signal, that's called a synapse. And so here's a picture of a couple of nerve cells. And you can see that these are both myelinated nerve cells, so they're very fast. And you can see that the signal always just moves in one direction. It never moves in the other direction. It always moves from the dendrites down to the axon. And so the signal moves from one nerve. The axon of that nerve, the axon end of that nerve, that whole thing is kind of called the axon, but the end of that nerve interacts with the dendrites of the next nerve, and that spot is called a synapse. And so that's how we can take the signal from one nerve and pass it to the other nerve. Okay, so in this figure from your book, we're going to zoom in on one of those synapses. So there's lots of these where, the nerve interacts with the next nerve. We're just going to look at one of them. We're going to zoom in on the membranes of both nerves, and that's the synapse. All right? And so that's figure 33.8 from your book. So the neuron that's bringing in the signal, that's called the presynaptic neuron, before the synapse. The opening between the two neurons is called the synaptic cleft. And then the neuron that's receiving the signal is called the postsynaptic neuron. So if you study these terms, they make sense. Okay, well, let's go through all these steps. So I'm just going to sit on this slide here, and we're going to go through these in order so we can understand what happens. So step one over here, the presynaptic action potential arrives at the synapse. So the sodiums are flowing in, and that signal gets all the way, now we're all the way to the synapse. Step two, the action potential causes an influx of calcium. So how? Well, the same way it's been propagating the whole length. Voltage sensitive channels. Except these channels are for calcium, not sodium. But that change in voltage causes those calcium channels to open, so calcium comes rushing in. Step three, calcium induces vesicular exocytosis and acetylcholine is released. Okay, that's a mouthful. What does that mean? It induces vesicular exocytosis. Go back to basic cell biology. Vesicular, a vesicle, right? A little bubble that contains something. Exocytosis. Exocytosis is when a vesicle binds to that membrane and fuses with that membrane and then releases whatever's inside the vesicle, releases it outside the cell. And so the calcium induces vesicular exocytosis, means you've got these vesicles that contain something. When calcium gets there, that stimulates them to go bind to the membrane of the neuron and release whatever they were holding. Well, what were they holding? In this example, it's acetylcholine. Acetylcholine is what we call a neurotransmitter, and there's lots of these, but acetylcholine is a very common one. So you got a vesicle of acetylcholine. When calcium comes in, that stimulates that vesicle to bind to the membrane of the presynaptic neuron, the one that's carrying the signal, and it releases that acetylcholine into the synaptic cleft. So that signal's moved all the way down this neuron, and the end result is to release acetylcholine. Now that neuron is done. It's wiped its hand, it's going to repolarize, and it's ready to do it again. Okay, step 4. Acetylcholine binds to a receptor, causing chemically gated ion channels to open and postsynaptic membrane excitation. So again, you can read that through and break it down. But the acetylcholine binds to a receptor. So in the postsynaptic neuron, the neuron that's receiving the signal, it's got receptors, it's got proteins on its membrane surface that are perfectly shaped for acetylcholine. And so the acetylcholine can bind to those receptors. What's that do? Causes them to change their shape. And so causing chemically gated ion channels. So when a chemical binds, it opens the gate and it allows ions to flow in. And so it causes chemically gated ion channels to open and postsynaptic membrane excitation. Well, what kind of gates are these? If we're going to excite this nerve, what do we need to do to get this nerve excited so that it will form an action potential? We need sodium to rush in, right? And so how do we start that signal? This is one way to start that signal. When acetylcholine binds to its receptor, that opens up a sodium channel, allows sodium to rush in, that excites that neuron. And so number 5, postsynaptic excitatory potential. So that sodium starts to rush in. Now the postsynaptic, the new nerve, starts to get more positive inside the cell. Number 6, postsynaptic action potential if enough excitatory potentials are received. And so if enough sodium rushes in to raise that inside voltage to plus 35, then that opens other sodium channels, and then we just repeat the process. Now the signal just travels down this new neuron if enough sodium rushes in. So if only a couple of acetylcholines bind and only some sodium rushes in, it's not enough to open other channels, and so the nerve won't fire. But enough of them open up, enough sodium rushes in, the voltage changes enough to open other nearby sodium channels, then they allow sodium in, which changes the voltage and opens nearby sodium channels, and then boom, you got another action potential in this new nerve. Okay, so at that point, you have passed the signal to the other nerve. And that nerve, if you get enough sodium, that nerve will fire, and then the signal just keeps moving on. And that's what we want. Now, number 7, acetylcholine inactivated by acetylcholinesterase into acetate and choline. What's it saying there? The acetylcholine is broken down. That's all it's saying. So the acetylcholine was bound to this receptor, which caused sodium to fly in. Now, acetylcholinesterase, right, it's an enzyme, breaks down acetylcholine, and so then it's no longer bound to its receptor, so the gate closes back up. So now sodium doesn't rush in anymore. So those two pieces of acetylcholine, step 8, choline is reabsorbed into the presynaptic terminal. And so one part, part of that acetylcholine gets reabsorbed back into the original neuron, into the presynaptic neuron. Number 9, acetylcholine resynthesized from acetate and choline is stored in vesicles. So you got part of that acetylcholine comes back in, you grab some other stuff, you put them back together, you make a new acetylcholine molecule, you put it in a vesicle, and now you got a vesicle of acetylcholine. And the next time this nerve fires, calcium will come in, that vesicle will bind, release the acetylcholine, and we repeat. So this is a synapse, and this is how nerves can pass their signal from one nerve to the other. All these little chemical steps here. And there's a lot of these neurotransmitters. There's a lot of different things going on here. This is the 10,000 foot view, but you get the basic idea here of, again, it's just, ion channel opening and letting ions flow in and then closing those channels. That's how we're controlling this whole thing. We're just doing it chemically here. And so those neurotransmitters carry that signal across the synaptic cleft to the next neuron. And so some synapses, when those neurotransmitters go across that synaptic cleft, that opens sodium gates. So that's what we just saw, where you're opening sodium gates, and it makes it more likely that the second neuron will fire because you're letting sodium in. Those are excitatory synapses, right? Like we're opening sodium gates, we're letting sodium in. If we let enough sodium in, then that will trigger other sodium channels to open and the nerve will fire. And that's the example we just saw. But like I said, there's lots of synapses, lots of neurotransmitters. There are other transmitters, other synapses, which might have different neurotransmitters, might open other gates, like chloride gates, for example. And so that's going to make that postsynaptic membrane less likely to fire. Remember, if you let chlorine in, the membrane becomes hyperpolarized, and so it's harder to get the voltage needed to start an action potential. And so other synapses might do that, and those are called inhibitory synapses. And so you've got some nerves that are excitatory and some nerves that are inhibitory, and they all kind of interact with other nerves, and they all kind of fight it out, and the net result determines whether or not that postsynaptic nerve fires or whether it doesn't fire. And so that's what kind of showing you on this figure. You've got lots of synapses coming in to each neuron. And so if the excitatory ones win, if enough sodium channels get opened, then enough sodium gets in, then the postsynaptic neuron will fire. If the inhibitory ones win, meaning that you get more negative ions or whatever, and so you don't get enough sodium to cause other sodium channels to open, then that postsynaptic nerve won't fire. And these chemicals are constantly battling it out, and the net result is either the nerve fires or it doesn't fire. So previously I asked what starts all this? What opens that first sodium channel, which can trigger other sodium channels? Well, this is one way. This is not the only way, but this is one way to open that first sodium channel. A signal comes from an excitatory neuron, which releases neurotransmitters, which open enough sodium channels to start this action potential. Wow, that's a lot, all right? But I want you to go back and study those figures. They're really good figures. And if you kind of go through the steps, it all makes a lot of sense. And so then in future lectures, we're going to talk about the end result of this and what this means for neurons firing and things. But just like with the contracting of a muscle, you know, we've dialed in on a little bit of detail, but now we can really understand this a lot more deeply. So anyway, I think it's cool. You let me know if you got any questions, and I'll see you in a bit. Later.